Hello students, thank you for tuning into this lecture on Chapter 5. This is not the entire Chapter 5 lecture, but I have just decided to record a lecture on a few of the important points that you may deem as necessary to go over. We're going to pick it up here from the generation of ATP slide. Most of the energy released during oxidation reduction reactions is trapped within the cell by making ATP. This symbol, I want to point out this particular symbol here, okay? This symbol with the little squiggly line indicates a high energy bond. When the phosphate is removed, energy is released. The energy is in the bond, so when the phosphate is broken off, energy is released. The addition of a P of a phosphate to a chemical compound is called phosphorylation. The addition of the P of the phosphate can take place in three different ways. One way is through substrate level phosphorylation. The term substrate should automatically imply that an enzyme is being used to phosphorylate ADP. The PO4 is the oxidized form of a phosphate, and it is a phosphate ion, which is also inorganic, by the way. So ADP, D stands for diphosphate, which is two phosphates. The P is attached to the ADP, creating ATP, and ATP, the T stands for 3. So now there are 3 phosphates here, which means that this molecule now has more energy contained within it. Another way that ATP is generated is through oxidative phosphorylation. In oxidative phosphorylation, electrons are transferred from organic compounds to a group of electron carriers. These electron carriers are NAD+, and we also have FAD. The transfer of electrons from one electron carrier to the next releases energy, some of which is used to make ATP through a process called chemiosmosis. This will be described later. Lastly, another way to make ATP is through photophosphorylation. Photophosphorylation will imply that light is being used, and this occurs only in photosynthetic cells. The photosynthetic cells have light-trapping pigments called chlorophyll, which release electrons when it's energized. In this lecture on microbial metabolism, we are going to start off with talking about carbohydrate catabolism, basically the breakdown of carbohydrates and how this is used to make energy. Organisms release and store energy from organic molecules by a series of reactions that are very controlled. The organism does not release the energy all at once. And the reason for that is because there will be too much heat that is released, because every time there's an energy conversion, heat is released. And so that will be way too much heat. So instead, the carbohydrates are broken down in 3 different steps here. And within these steps, within these stages, you would still see a step-by-step breakdown of the molecules. First here we have glycolysis, which means the splitting of sugar. Then the second step is the Krebs cycle, and then we will look at the electron transport chain, all of which will generate energy in some form. And right here I would like to add that there are 2 general processes that are used to produce energy from glucose. One is cellular respiration, which we will now go about speaking of, which would include the glycolysis, the Krebs cycle, the electron transport chain. And then there's also fermentation, which we will discuss towards the end of this lecture. Both cellular respiration and fermentation start with glycolysis, but they follow different pathways. So now let's talk about cellular respiration. So most of the microorganisms oxidize carbohydrates as their source of cellular energy. So we are going to start off with the carbohydrate catabolism, which is a breakdown of carbohydrate molecules to produce energy. Glucose is the most common carb energy source used by cells. Microorganisms can also metabolize other biomolecules like lipids and proteins, but we're going to start off with the process looking at the catabolism of glucose. The first stage of carbohydrate catabolism would be glycolysis. And glycolysis means the splitting of sugar. Glyco refers to sugar, and we have the lysis, which refers to the splitting or the breaking apart of sugar. So, glycolysis means splitting of sugar, and this is exactly what this process does. It splits the sugar, which is a 6-carbon molecule, into 2 3-carbon molecules of pyruvic acid. Now, I'm going to have to ask you from now, and you've heard this in other lectures of mine, that to please forgive me for the formatting. For whatever reason, the app that I use to make these do not always format the slides properly, but hopefully you can follow along if you have the slides in front of you that I provided on our website. So the 6-carbon molecule here, which is glucose, you can see all we're illustrating are the carbons, which are these gray balls. The 6-carbon molecule is split apart into 2 3-carbon sugars that are oxidized, and they release energy. Their atoms are rearranged, forming 2 molecules of pyruvic acid or pyruvate. During glycolysis, NAD plus is reduced to NADH. So when we have this splitting take place here, the electrons that are released will jump into the NAD, which NAD plus, by the way. And once it jumps into NAD plus, this means now that it's gained, so that would be a reduction, and it is reduced to NADH. There is a net production of 2 ATPs by substrate level phosphorylation, so an enzyme is used to make it. And here you can see that glycolysis did not require any oxygen whatsoever. So glycolysis can occur whether oxygen is present or not. The presence of oxygen does not stop this from taking place, but it can occur in any situation. To get this reaction started, I want to point out that it requires 2 ATPs going into this reaction to get the whole ball game rolling here. But altogether, 4 ATPs are made, but because we have to pay back those 2 ATPs that were initially put in to get the ball rolling, this is why we say that we only have 2 ATPs that come out of glycolysis. So the diagram that we just looked at was highly simplified. Glycolysis is actually a series of 10 chemical reactions. And here we have the first half of the steps outlined. So first of all, in step 1, what we see here would be that glucose enters the cell and it is phosphorylated. So the 2 ATPs, 1, 2, attach their phosphates to the glucose molecule. And now that it's highly energized, it is going to basically rip that 6-carbon molecule apart to make 2 3-carbon molecules, So in step 1, the glucose enters the cell, it's phosphorylated, and then the glucose 6-phosphate is now rearranged in step 2 into fructose 6-phosphate. Then, the other P will be attached now from the second ATP. And then we will have the sugar splitting into 2, an enzyme and a phosphate having the high energy make it very unstable. And that's why it breaks into the 2. And then now we're going to have DHAP, which is readily converted into GP. You can see that this is a reversible reaction. On this slide, I want to remind you guys that on the previous slide, we ended off with a GP, which was a 3-carbon compound. So now picking up from this slide, which starts here at step 6, we have an enzyme in the cell that will convert that GP that we just ended off with to another 3-carbon compound, which is called 1,3-diphosphoglyceric acid. Now, in this, the intermediates are really not that important. I'm not going to require you to remember all of those intermediates, but I do want you to be able to understand a general idea of what's going on. When that GP got turned into this 1,3-diphosphoglyceric acid, there would have been, the GP was oxidized, which means that something has to get reduced. And what got reduced would have been this 2NAD plus, which got reduced into NADH, and we will then see this NADH, the energy, get harvested in the electron transport chain later on. The enzyme couples that reaction with the creation of a high energy bond between the sugar and the phosphate, which you can see right here. Remember, the squiggly line would be a high energy bond. Then in step 7, the high energy phosphate is moved to an ADP to form ATP. This step is going to repay the initial investment of the 2 ATPs. Remember, even though I only have one set of reactions being shown here, please remember that you do have 2 of these reactions because that molecule got broken apart into 2 3-carbon compounds. Here I'm only showing 1. So an enzyme will now come and relocate the remaining phosphate. It relocates it right here in this step that you see as step 8. It relocates that remaining phosphate in preparation for the next step. In this, that will cause water to be lost. The loss of water causes that phosphate bond to be upgraded to a high energy bond, which is why now you see the squiggly line. This the phosphate is transferred to an ADP to make a TP. This now leaves us with a 3-carbon molecule that we call pyruvic acid. After glucose is broken into pyruvic acid, the pyruvic acid can be channeled into either fermentation or it can go into the next step of cellular respiration. Some bacteria have another pathway in addition to glycolysis for the oxidation of glucose. We have the PPP, which is the pentose phosphate pathway, and this uses pentoses, which are 5-carbon sugars, as well as glucose. You see here that it operates with glycolysis. Organisms like B. subtilis, E. coli, E. faecalis, these will use the PPP, and they produce a lot of important intermediates that are used in the synthesis of nucleic acids and amino acids. Then we also have the EDP, which produces, from each molecule of glucose, 2 NADPHs are made here, and we also have an ATP that's produced. Bacteria that have the enzymes for the ED pathway can metabolize glucose without the use of glycolysis. So remember, the first step of cellular respiration was glycolysis, where sugar got split into 2-pyruvic acid. But not all of the energy that was in that glucose was extracted. We did get energy in the form of 2 ATPs, and we also got NADH, but there's still a lot of energy in the pyruvate. So you're going to see that most of the energy will be generated in oxidative phosphorylation, which is in the last step of cellular respiration. This is where we will get all of the energy out of the NADPHs and the FADH. These will get energy out of those electron carriers. Well, in that case, they're reduced, so they have tons of energy in them waiting for us to extract. Between glycolysis and the Krebs cycle, there is a step called the intermediate step that chemically grooms the pyruvic acid. Pyruvic acid cannot enter the Krebs cycle. So in its preparation, it loses one carbon. So here's that pyruvic acid that got made in glycolysis. It loses one carbon in the form of carbon dioxide. And this is called a decarboxylation. The intermediate step further grooms this molecule by attaching a coenzyme A, to the two carbons that are left. And this is now called acetyl-CoA. Now, the molecule can be moved into the second step, which is the Krebs cycle. So, leaving off from the step before, we remember that the pyruvic acid was groomed into acetyl-CoA, and the whole point of that coenzyme is strictly to bring the molecule into the Krebs cycle. Once it's done that, it detaches itself and then it goes back to pick up it goes back and it's used again to groom another pyruvic acid molecule to get into the Krebs cycle. As acetyl-CoA enters the cycle here, so it will enter at 1. Remember that it has 2 carbons. So we're going to be able to follow the carbons in this Krebs cycle. Because it's a cycle, that means that it has to regenerate its starting material. And the starting material is the oxaloacetic acid. This 4-carbon molecule will join with the 2-carbon molecule that is coming from the acetyl-CoA, and that will make a 6-carbon molecule, which we now see right here, which is the citric acid. So the Krebs cycle is also called the citric acid cycle, and that's where it gets its name from. Now, in step 2, what we see is a rearrangement of this 6-carbon molecule. It is rearranged into isocitric acid, which is also a 6-carbon molecule. In steps 3 and in steps 4, we will see the carbon being released in the form of carbon dioxide. So this is a decarboxylation, but here you can see the carbon dioxide, carbon being released each time. So these oxidations are going to generate NADH. So just to kind of walk you through, here is the 6-carbon isocitric acid. A carbon is removed in the form of carbon dioxide, which will now make this 5 carbons. When that happened, though, the electrons that were released jumped onto this NAD+, forming NADH. Now that is going to go over to the electron transport chain. Next, this ketoglutarate acid, the alpha ketoglutarate, which was 5 carbons, gets another carbon dioxide stripped off of it, another carbon stripped off of it in the form of carbon dioxide, which will now leave us with a 4-carbon molecule that you see here. In the process, the electrons that were released with the stripping of that carbon jump into the NAD+ here, thus reducing it to NADH, and we will see that later on in the electron transport chain. ATP is also produced here in the at substrate level phosphorylation, so an enzyme is used. In steps 6 and 7 and 8, enzymes rearrange the chemical bonds here, and it produces 3 different molecules before generating, or maybe I should say regenerating, oxaloacetic acid. Through steps 6, 7, and 8, there are important energy-rich molecules that are being made. We do have NADH being made again, which will go off to the electron transport chain. And then here we also see FADH, and that will go to the electron transport chain. So in the Krebs cycle, we have showing here 3 NADHs. We also are seeing 1 FADH. But remember that there are 2 pyruvates coming in, 2 of these. So everything needs to be multiplied by 2. So it's really 6 NADHs and 2 FADHs made in the Krebs cycle. Now that we've gone through glycolysis and the intermediate step and the Krebs cycle, we are moving towards the very last stage, which involves the electron transport chain. And in the electron transport chain, there are a series of molecules that are carrier molecules. They carry electrons, and there are a lot of oxidation and reductions that's going to be taking place as the electrons are passed down the chain. The electrons are going to be passed down the chain to oxygen. In cellular respiration, oxygen is the final electron acceptor. And the energy that's released as these electrons fall down to oxygen will then be used to make ATP by what we call chemiosmosis. So this slide is simply showing the electron transport chain having a sequence of carrier molecules that are being shown here. And these are capable of oxidations and reductions. When electrons are passed down this chain to the oxygen molecule here, then energy is going to be released in a stepwise fashion. And this energy is going to be used to drive the chemiosmosis, which makes this ATP. Now, remember what I mentioned in the beginning that when the organisms are generating ATP, they start off with glycolysis, and it can move in any direction as far as the other steps, which would be in respiration, you have glycolysis, then you will have the intermediate step, and then finally you'll move into the electron transport chain and chemiosmosis. Or it can take the other path here to fermentation, which will be discussed later on. At this point, we are now getting to this section right here, electron transport chain and chemiosmosis. Now I'm going to discuss how the cell uses the electron transport chain and an enzyme embedded within the membrane to make a whole lot of ATP. So finally, we are at the last stage here, chemiosmosis, where we will see most of the ATP getting made. That was most of the energy is finally going to be harnessed that was trapped in the glucose molecule that we started off with. So chemiosmosis, we see here the inner mitochondrial membrane. This is in eukaryotes. If we were looking at a prokaryotic cell, then we would be looking at the periplasmic space. Now in here, there are three classes of carrier proteins embedded in the membrane. First, we have flavoproteins. These contain flavin. And flavin is a coenzyme that's derived from riboflavin, vitamin B12. And these can perform alternating oxidations and reductions. Then there is a second class of proteins here that we call the cytochromes, which you see here, CYT. And these cytochromes are proteins with an iron-containing group. The third type of protein that you're going to see here will be here indicated by Q, and these are the ubiquinones or coenzyme Q, and these are non-protein carriers. The carriers used by bacteria and the sequence differ from one bacteria to another, but all of the electron transport chains, they achieve the same goal. They have the same goal in mind. So here what we see is, and I'm just showing NADH. Remember that there were also two FADHs made, but we're going to follow one molecule of NADH. So NADH moves in. Remember, these were made in the previous steps. One NADH moves in and it puts its electrons onto the first electron carrier that we see here. So this means that the NADH is oxidized into NAD+, and because that was oxidized, and these are couple reactions, something had to have gotten reduced, and what got reduced would be the very first protein here. So the NADH puts its electrons onto the very first protein. Now, what would happen is that the electrons are going to get shuttled down one protein at a time until it gets to the oxygen. Hopefully you remember that oxygen is electronegative. It attracts electrons to itself. So when the very first electrons get put on the first protein, it's just going to make its way down to that oxygen, down the electron transport chain. As that happens, a little bit of energy is going to be released. And that energy that's going to be released is now going to be used to shuttle the hydrogen ions here. You would either hear me call them protons or hydrogen ions. These would get shuttled against their concentration gradient. So they will be moving in this direction. And remember, they're basically going from low to high, which is not how things usually go. So this is going to require energy. And the way that it gets energy is once again through the fall of the electrons down to oxygen. As the electrons get passed from one protein to the other within the inner mitochondrial membrane, the energy that's released is going to be used to pump the hydrogen ions against the concentration gradient. Now, the last part here, though, is the actual making of the ATP. Now, as you can see here, the ATP is generated right in this, the ATP synthase. So this is an enzyme. It has the ASE ending, and this is what is going to be used to phosphorylate the ADP and turn it into ATP. So how does this happen? Well, the hydrogen ions, remember, these are responsible for the pH of substances. So when the fluid gets enough hydrogen ions, the acidity will increase. And at a given pH, the ATP synthase is going to be activated. Please recall that enzymes are sensitive to the environment. The pH will affect enzyme activity. So when the hydrogen ions are placed or put into this particular space, then at some point the ATP synthase will be activated and all the hydrogen ions will run through this ATP synthase, and it turns a little turbine within the ATP synthase, and that energy that is generated there will be used to phosphorylate the ADP with this phosphate, and it becomes ATP. One thing you'll also notice is that the NADH, for one NADH, you end up getting 3 of these ATPs. And for one FADH, we end up getting 2 ATPs. And this picture is only showing the NADH given 3 ATPs, but one FADH gives 2 ATPs. And the reason for that would be because when NADH comes in, it puts its electron onto the very first protein, whereas the FADH puts its electron to a protein that's further down on the electron transport chain. So the NADH is traveling much further down to oxygen versus the FADH, which does not go so far. And because potential energy is stored within an object based on its location, if the NADH is put on the first versus the FADH, which is put further down, then the NADH would end up giving out more ATP. It will be worth more energy. Overall, aerobic respiration makes about 36 ATPs in eukaryotes, which is fewer than in prokaryotes. Eukaryotes spend energy shuttling the electron carriers from the cytoplasm to the mitochondria, but prokaryotes do not have these specialized organelles, so they don't spend energy doing that. And in anaerobic respiration, it has a final electron acceptor, but it is not oxygen. The final electron acceptor in aerobic respiration is oxygen. And this should be a subscript, but again, remember our formatting is messed up here. The anaerobic respiration has an electron acceptor as anything other than oxygen. Some use nitrate ions like Pseudomonas and Bacillus. Those genres use nitrate ions. Some use sulfate, some use carbonate. The amount of ATP made in anaerobic respiration varies with organism and pathway, but it never is as high as aerobic respiration. This slide is simply showing the electron acceptors in anaerobic respiration. Remember, it's anything other than oxygen. So in this case here, we have a nitrate ion, and this one is the product would be a nitrate ion that's reduced to a nitrite ion. So here we have nitrite and nitrous oxide or nitrogen gas. Here we have the sulfate forming, eventually that forms the hydrogen sulfide over here. And again, remember, these are subscripts. And then here we have carbonate that forms methane. This slide is showing us the locations of each of the pathway where each is taking place in either the eukaryotic or prokaryotic cell. You can see here that glycolysis takes place in the cytoplasm for both prokaryotic and eukaryotic cells. The intermediate step, again, both in the cytoplasm for both eukaryotic and prokaryotic cells. In the Krebs cycle, that takes place in the eukaryote in the mitochondrial matrix. But remember that prokaryotes do not have these specialized organelles, so it takes place in the cytoplasm. And the electron transport chain is in the mitochondrial inner membrane, whereas the prokaryotic cells, it takes place in the plasma membrane because, again, they do not have a mitochondria. And this table summarizes the amount of energy that's produced from the complete oxidation of one glucose. So if we follow one glucose, what do we get out of it when we look at aerobic respiration? So remember, we had glycolysis, and that gave us 2 ATPs, but it also gave us 2 NADHs, which will be harnessed. The energy from this will be harnessed in the electron transport chain. Then we have the intermediate step. We didn't get any ATP produced, but we did get 2 NADHs, which will be harnessed in the electron transport chain. The Krebs cycle produced 2 ATPs, and we got 6 NADHs. Now, remember that the diagram that I showed you, it showed 3 NADHs and 1 FADH, but we times everything by 2 because it was 2 of those molecules that were coming in. So we got 6 NADHs and 2 FADHs. Remember that the NADH yields 3 ATPs because it's put higher up on the electron transport chain, and the FADH yields 2 ATPs because it is put lower down on the electron transport chain. Now, when we want to get the complete amount of ATPs that were made, if we look at glycolysis, it was 2. We've got intermediate step, which gave us no ATPs, but the Krebs cycle gave us 2. So altogether, that would be 4 ATPs that were used, that could be used for any cellular work. But we also had those NADHs and the FADHs. So from one molecule of glucose, we end up getting we had 2 NADHs made in glycolysis, 2 in the intermediate step, and we had 6 made in the Krebs cycle. So if you multiply all of that by 3, which is how many ATPs the NADH is worth, you'll see the various numbers resulting in 30 ATPs. And then in FADH, we got 2 in the Krebs cycle. So remember, we multiply that by 2 because FADH is worth 2. And now we've gotten 4 ATPs. If we put all of these together, this will give us 36 to 38 ATPs. 36 in eukaryotes because some energy is spent during the process, whereas 38 would be in prokaryotes because they don't spend that extra energy bringing in the electron, the NADHs and the FADHs into the mitochondria. So far, we've looked at aerobic respiration, which uses oxygen. After glucose has broken down into 2-pyruvic acid, the pyruvic acid can be completely broken down in respiration, as we have spent this entire time discussing, or it can be converted to an organic product in fermentation. So fermentation, it releases energy from sugars or other organic molecules such as amino acids, purines, or pyrimidines, which would be nucleotides. It does not require oxygen, but sometimes it can occur in its presence. It uses an organic molecule as the final electron acceptor. It does not use the Krebs cycle, or it does not use the electron transport chain. Because it does not use the electron transport chain, it only produces a small amount of ATP. So here we have the pyruvic acid, which is made at the end of glycolysis or during glycolysis. And we can see all of the different organisms here. Microorganisms can ferment various substrates. Here it is showing pyruvic acid. We see the end product is different depending on the microorganism. The substrate and the enzymes that are present and active is what will determine the end product. The analysis of the end products are useful in helping us to identify an organism. Next, we're going to consider two important processes. We're going to consider lactic acid fermentation and alcohol fermentation. Okay, so here we have alcohol fermentation, and this begins with glycolysis and produces ethyl alcohol, and it produces carbon dioxide. These waste products made by yeast are useful to humans. Ethanol made by yeast is the alcohol in alcoholic beverages, and the carbon dioxide causes bread to rise. Lactic acid fermentation starts with glycolysis as well and forms two molecules of ATP. So we get ATP out of this. Two important genera of lactic acid bacteria are Streptococcus and Lactobacillus, which we have run into in the lab. They produce only lactic acid and are therefore referred to as homolactic, meaning that they produce only lactic acid. Organisms that produce lactic acid as well as other acids or alcohols are considered heterolactic. In this diagram, we have lactic acid fermentation and alcohol fermentation illustrated. In lactic acid fermentation, we're going to look at these starting from pyruvic acid, since you already know how glycolysis takes place. Now, in lactic acid fermentation, two molecules of pyruvic acid are reduced by two molecules of NADH to form two molecules of lactic acid. So here's the pyruvic acid reduced by the NADH to form 2 lactic acid. Lactic acid is the end product and undergoes no further oxidation, and most of the energy produced by the reaction remains stored in lactic acid. There's a very small yield of ATP. It is essentially the 2 ATPs that were made in glycolysis. But it's enough for the microorganisms' needs. The question here is, why continue to process this pyruvic acid if the ATP is already made? We've already gotten ATP out of this, so why continue to process the pyruvic acid if we're not going to get any more energy? And really, it's just so that we can regenerate NAD+. Why is it important to regenerate the NAD+? Well, that's because it's a reactant in glycolysis. If we don't have it, then glycolysis can't take place and the organism cannot make its 2 ATPs. Now, when we look at the pathway of alcohol fermentation, you would see that the pyruvic acid is converted into acetaldehyde, and it's also converted into CO2, carbon dioxide, which leaves here. The 2 molecules of acetaldehyde are reduced by 2 NADHs to form ethanol. And again, it regenerates this NAD+ so that it can do this reaction all over again. This is needed in glycolysis. In the lab, we will do several fermentation tests. This picture shows the results of fermentation tests. The test medium has protein. It also has a single carbohydrate, which is used as the energy and carbon source. And there's a pH indicator, which changes colors. There's also a Durham tube that you can see in here that is flipped upside down in here. And this captures gas, which is produced by some organisms. That gas, if you think about this, the holes that's in Swiss cheese, the holes that are in Swiss cheese are formed because of the accumulation of carbon dioxide gases. So here in this one here, in A, we have an uninoculated fermentation tube containing the carbohydrate mannitol. The sugar here is mannitol. In B, this was inoculated with staph epidermidis, and it grew because of the protein in the media, but it did not use sugar. So this is why the color stayed the same. Okay, so this one here did not use sugar. In C, we can see Staph aureus. This produces acid, but it doesn't produce gas. So we know that it produced acid because of the color that it turned, but there is no bubble. So we know that it did not produce gas. And here, in D, E. coli is positive. This one is E. coli in D. And we know that it's positive. It's used because, well, it's positive for acid because it changed color. And it's also positive for gas because there's a bubble that's trapped up in the inverted Durham tube. So far, we have emphasized energy production from glucose, which is the main energy supplying carbohydrate, but microbes can also oxidize lipids and proteins as well. Remember, fats consist of glycerol and fatty acids, and the microbes can produce enzymes called lipases that break down fats into these monomers. So it breaks it down into the glycerol and to the fatty acids. Each component is metabolized separately. The Krebs cycle functions in oxidizing glycerol and fatty acids. Bacteria that break down fatty acids can use the same enzymes to degrade petroleum products. This is a nuisance if they grow in a fuel storage tank. But what do you think these can be used for? They can be used to treat oil spills. So that's a wonderful way to help out here. We call it bioremediation. In addition to the sugars and the fats, proteins can also generate energy as well. But they are too large to pass through the plasma membrane without any help. So microbes produce extracellular proteases that will help to break down these proteins into their component amino acids. And then these amino acids can cross the membrane. Before the amino acids can be used, they must be enzymatically converted to other substances that can enter the Krebs cycle. So deamination, this is a process where the amino group of the amino acid is removed and converted to an ammonium ion, which can be excreted from the cell. Decarboxylation removes a carboxyl group, and dehydrogenation removes the hydrogen. In the lab, we will use tests that can detect amino acid catabolizing enzymes. Here we have a medium with glucose. It also has a pH indicator and a specific amino acid in it. In this first one, we'll call this one A. What we have here is the pH indicator turning yellow when bacteria produced acid from glucose. So this one shows acid was produced. And in this one, which we'll call B, we have alkaline products produced. And so these alkaline products were produced because of decarboxylation, and it turns the indicator purple. So these are some tests that we can use it to help us identify certain bacteria in the lab. So far, the metabolic pathways we have discussed involved organisms obtaining energy from organic molecules. But some organisms make complex organic compounds from simple inorganic substances in a process that we call photosynthesis. Photo means light, synthesis means making. This is carried out by plants, and it is also carried out by many microbes. So photosynthesis is the conversion of light from the sun into chemical energy. The chemical energy is used to convert carbon dioxide from the atmosphere to a reduced carbon compound. This reduced carbon compound will be sugars, and this is what we call this process carbon fixation. we say that it is fixing carbon into organic molecules. Photosynthesis can be summarized with the following equations. We have oxygenic, which is where plants, algae, cyanobacteria use water as a hydrogen source, H2O. Remember the subscripts. It uses it as a hydrogen source to donate hydrogen ions, and in the process, it releases oxygen. Then we have anoxygenic, which a lot of the purple and green sulfur bacteria use. They use hydrogen sulfide as a source of hydrogens to reduce carbon dioxide into sugar, which is right here. Again, reference your slides that are in front of you because these slides, the formatting has been completely messed up. None of the subscripts are evident. Photosynthesis takes place in two stages. We have the light-dependent reactions, and then we have the light-independent reactions. Sometimes people call the light-independent reactions the dark reactions, which I do not favor because it kind of indicates, it makes you think that this particular reaction has to happen in the dark, but it can happen in the light, it just does not use it. So it's light independent. We also call it the Calvin cycle or the Calvin-Benson cycle. So these are the two reactions that we're going to look at. In cellular respiration, there were three. There were glycolysis, Krebs cycle, and then we looked at the electron transport chain and chemiosmosis. Now we're going to look at two, which is the light-dependent reactions and the Calvin cycle. So when we look at photosynthesis, what you're going to see is that chlorophyll molecules are involved. So here we have chlorophyll. And these absorb light energy. Here's the light coming in. And all of the electrons that are on the chlorophyll molecule, these get excited. When they get excited, they jump from the chlorophyll. So they fly up here and they jump to the first series of carrier molecules, which is in an electron transport chain. And hopefully you understand the electron transport chain because there was one in cellular respiration. So they jump up and they make their way down the electron transport chain. The energy from the electron transfer is used to make ATP by chemiosmosis. Now here we are showing cyclic photophosphorylation because the electrons that are released from the chlorophyll by the light eventually will go down the electron transport chain, but they are returned to the chlorophyll after the passage along the electron transport chain. So because they are returned right back to the chlorophyll, we call this cyclic photophosphorylation. There's a cycle of the electrons. Now, there's also non-cyclic photophosphorylation, and this is used in oxygenic organisms, the organisms that use water as a source of hydrogens. So the electrons are released from the chlorophyll, but you would see that, yes, they get excited, they jump up, they go down the electron transport chain, releasing energy to make ATP, just like in cellular respiration, but instead of the molecules coming back in cyclic photophosphorylation, those electrons will be returned to the chlorophyll molecule. But in non-cyclic, this does not happen. So what we have here is the electrons being passed to NAD plus, NADP plus, and reducing it to NADPH. So here the electron acceptor is NADP plus. Now the electrons that are lost from the chlorophyll molecule are simply replaced by the water, by the splitting of water. And this is where oxygen is made. When water gets split apart, the hydrogen ions are here, the electrons are put onto the chlorophyll, and the oxygen is released. So that first portion would have been the light reactions because you saw light being used in the cyclic and non-cyclic photophosphorylation. But now the Calvin cycle, what we call the dark reactions, does not use light. It doesn't use it, but it can happen in the light. And this is where carbon is fixed into organic materials. Here we are going to use 18 ATPs. Of course, it's not shown necessarily like the 18. It shows 6 here, but altogether it will be 18 ATPs because this cycle turns around 2 times. So if we have 6 here plus 3 here, that's 9 ATPs, but we multiply it by 2. So we get 18 ATPs that are being used here. And we also have NADPH coming in, and this is being used. We need it for its reducing power. And again, remember, this is 2 rounds, produces a glucose. So this has to go around twice. In the first step, 3 carbon dioxide molecules from the air are attached to 3 5-carbon ribulose diphosphate here. This is a cycle. So this material will join with the 3 carbon dioxides coming in. And when this happens, they will react with water and break into 2 3-carbon molecules. This is how the carbon, though, in this stage, we call this carbon fixation because now the carbon has been put in to this molecule. Now, we can also see here that the ATPs are used to phosphorylate the 3-phosphoglyceric acid, and it turns it into 1,3-diphosphoglyceric acid, which gets reduced by NADPH, turning it into G3P. After G3P is produced, 1 out of every 6 is taken out of the cycle. When 2 are taken out, they react to make other sugars and glucose. So you'll see here glucose and other sugars. 5 out of every 6 G3Ps continue in the cycle, so that the RUBP right here can be regenerated, because this is the starting material for carbon fixation, so that when carbon dioxide comes in, it can be joined to the RUBP and start this entire cycle all over again. This table compares photosynthesis in eukaryotes and prokaryotes. Remember that the green and purple bacteria are anoxygenic, but here you can see the characteristics. So please look over this table, and you can see that the eukaryotes, for example, use hydrogen atoms in H2O to reduce carbon dioxide. Cyanobacteria does the same thing because those are oxygenic. But in prokaryotes, the reduction power basically comes from sulfur compounds or sulfur. You can also see that the oxygen production, eukaryotes are oxygenic, cyanobacteria oxygenic, but anoxygenic for green and purple bacteria. The type of chlorophyll that's used here would be chlorophyll A, cyanobacteria chlorophyll A, but the green and purple bacteria use bacterial chlorophyll A or B in the purple bacteria. The site of photosynthesis would be chloroplast with the thylakoids in algae and plants. Cyanobacteria would be the thylakoid. The green bacteria uses the chlorosomes, and chlorosomes are folds in the plasma membrane that contain bacterial chlorophyll. And the environment in which photosynthesis takes place for the eukaryotes or cyanobacteria would be aerobic, and it could also be anaerobic for cyanobacteria. And for the purple and the green bacteria, these are anaerobic. Halobacterium, which is a class of Euryarchaeota, these use bacterial rhodopsin, which is a protein, again used here by archaea. So all of these different colors that you see are archaea, and the bacterial rhodopsin act as a proton pump, and it captures light energy and uses it to move protons across the membrane out of the cell. The proton gradient that is created is converted into chemical energy. So here again, it's not chlorophyll that's used, it is bacterial rhodopsin, which is a protein. So we have looked at energy generating pathways in detail. We can classify organisms metabolically according to their nutrition pattern, meaning that, we're looking at their source of energy and carbon. Considering energy source, we can generally classify organisms as phototrophs or chemotrophs. Chemotrophs are organisms that get their energy by the oxidation of electron donors in their environments. So, chemoheterotrophs use the electrons from hydrogen atoms in organic compounds as their source of energy. Then we have chemoautotrophs. These use electrons that come from reduced inorganic compounds as a source of energy. Carbon dioxide is their main source of carbon. Inorganic sources of energy include sulfur, ammonia, nitrite ions, or here we have thiobacillus ferroxidans using ferrous iron. The oxidation of these inorganic compounds is stored in ATP, which is produced by oxidative phosphorylation. We have phototrophs that use light energy, and photoautotrophs use energy in the Calvin cycle to fix carbon dioxide. We also have photoheterotrophs. These use light source as energy, but they cannot convert CO2 into sugar. So for their sources of carbon, they use alcohols or fatty acids or other organic compounds and carbohydrates. The green and purple non-sulfur bacteria are photoheterotrophs. They are anoxygenic. This slide simply shows the diversity among organisms as far as their nutritional types, whether they're photoautotrophs, photoheterotrophs, chemoautotrophs, chemoheterotrophs, their energy source, whether it's light or chemical. You also have their carbon source, whether it's carbon dioxide or organic compounds. And here we have some examples of each. So be sure to go over these and understand, at least know these. Now, let's talk about how cells use ATP pathways for the synthesis of organic compounds such as carbohydrates, lipids, proteins, and nucleic acids. So we have polysaccharides, and these are big sugars. The carbon atoms required to make glucose are derived from the intermediates made during glycolysis and the Krebs cycle. After making glucose, the bacteria assemble it to make more complex sugars such as glycogen. UTP is uridine triphosphate, and this is an energy source. Right here, you see, anytime you see that big star, you know that we're talking about energy. And we also have things like UDPH, UDPNAC. These are all forms of sugars. They're used to make sugars. Now, lipids are made, they vary in their composition, so they're made in different ways. Cells make fats by joining fatty acids and glycerols. So you see a glycerol, you have fatty acids, you put that together to make some lipids. Glycerol comes from an intermediate during glycolysis. And you can see here glycolysis, glucose is broken down into this G3P here, and glyceraldehyde 3-phosphate. And This is an intermediate in glycolysis, which is used to make glycerol. Fatty acids built from two molecules of acetyl-CoA are added to each other. So here's the acetyl-CoA that is made when the coenzyme A is attached to the 2-carbon molecule. When the pyruvic acid had the carbon dioxide stripped off, and then the coenzyme A attached to it, we got the acetyl-CoA. The fatty acids are built from the two molecules of acetyl-CoA, and then they're added to each other. This requires energy and is not always in the form of ATP. To make proteins, amino acids are required because these are the monomers of protein. Some microbes, like E. coli, already have the enzymes present to allow them to use starting materials of glucose and also inorganic salts to make the amino acids. Some organisms require the environment to provide them with these amino acids. In the Krebs cycle, an amine group can be added to pyruvic acid or to an organic acid, and it converts the acid to an amino acid. This is called amination. If the amine group comes from an existing amino group, then it is called transamination. So essentially, the transamination is we have new amino acids made with the amine groups from old amino acids. And this is a really good website. I can't show it here, but this is a good video, rather, talking about what transamination actually is. If you want, I think you can start it at 305, and it should show you transamination. DNA and RNA, these are made from nucleotides, which can consist of a purine or pyrimidine, and it also have a 5-carbon sugar and a phosphate group. The 5-carbon sugar comes from PPP or EDP, and hopefully you remember that these are some alternate pathways. And certain amino acids made from the intermediates in Krebs cycle and glycolysis participate in the synthesis of purines and pyrimidines. The nitrogen atoms from the amino acids form the purine and the pyrimidine rings, and the energy for synthesis is provided by ATP. We're going to end this just mentioning amphibolic pathways, which are metabolic pathways that have both catabolic breaking down and anabolic building up functions. If you think of the Krebs cycle, it helps with the breakdown of glucose, which is catabolic, but it also produces intermediates that can be converted into amino acids, which would be anabolic. Amphibolic pathways enable simultaneous reactions to occur, and metabolism is just integrated within this. And the double arrows will show the amphibolic pathways, as you can see here, circling a few. So again, the amphibolic pathways allow for both anabolism and catabolism. So this wraps up the lecture on metabolism. We talked about in detail cellular respiration, and then of course we talked about how we make organic molecules through photosynthesis. So make sure that you guys contact me if you have any questions about anything. If I need to clear anything up, I'll be happy to do so. In the meantime, happy studying.